Hello, my name's Mrs. Tyler, and we're going to do some biology revision today where we're going to draw along a knowledge organiser looking at sampling. Sampling is a technique used by biologists using a quadrat to estimate populations, and it comes up loads in the exams. So today's knowledge organiser looks a bit like this. So you need a piece of A4, you need it in landscape. We're going to look at competition, then we're going to look at using a quadrat with random sampling. That's by far the most common exam question. But then another type of technique, which is called using a transect. Right, let's get started. So today we're talking about measuring populations, but I'm not going to put my title in the middle just yet because I'm going to give this one a bit of a different layout. So as usual, we're going to plan out our knowledge organiser. We're going to make it about a third down here. And again, about a third all the way along the bottom. So we've got a long, thin section on the bottom, a short, thin section to the left, and a bigger, squarer section to the right. In this section, we're going to make some notes on populations. In this section, we're going to make some notes on random sampling. And in this section, we're going to make some notes on transects. So this whole knowledge organiser then is about using quadrats to measure populations. So I'm going to call it measuring populations. But by that, I mean using quadrats. So I'm going to put my title up at the top. All right then, so under populations, we're just going to make sure that we remember all the key terms. So the words we need to know are habitat, which, as you know, is where an organism lives, where it lives. We need to know the term population, which is the number of individuals of one species, the number of one species. So all the oak trees and all the beech trees and all the pine trees, not the total number of trees, species of trees. Community then means all of the species in a habitat or in an area. So that would be all the oak trees and the beech trees and the pine trees, but also all the other animals and plants that live there. So the owls and the mice and the bracken and anything else that might live in that habitat. Two more words we need to know are biotic. Bio means living. So biology is the study of living things, and biotic means living factors. So that's things that are alive that affect other organisms. So if biotic means living, abiotic means non-living. We also need to have a little think about competition. And that's when organisms are after the same resources. So if I'm drawing a plant, there's my soil, here's my roots, and we've got our light with our leaves capturing it. So if I've got my light over here, this plant is in competition for that light. It's also in competition for space. And it's also in competition for water and mineral ions. Remember, we say minerals from the soil, not nutrients. Mineral ions in the soil. And also water. There's also competition from other species of plants. So if this plant happens to grow next to, unfortunately, a taller plant, that might pinch the light and put the other one in shade. Right. So plants can have competition from abiotic factors, but they can also have competition from biotic factors, another living thing. So competition between another plant would be a biotic factor, because that is blocking the light. The light itself is abiotic, but the plant that's grown there blocking the light from reaching that one's leaves is biotic. All right then. So if we want to find out how many plants are growing in an area, then we need to count them, obviously. But there are a couple of obvious problems with that. The first problem is that plants are often very small, so there's very high numbers of them. And another problem is that they often grow in clumps. So plants can be small and grow in clumps. So this makes them hard to count. So instead, what we do is we take a sample. We don't count them all, we count a few. But to make our sample accurate, we need to do lots of repeats. So for random sampling, we're going to say that we need to do at least 10 repeats to make an estimate accurate. Repeat 10 times to calculate an accurate mean. Another thing, because they grow in clumps, which means you might have loads in one area and none at all in another area, you need to make sure that your estimate is representative. So you need to make sure that you're doing a bit of the whole area. So to do that and to remove bias, we need to do random sampling. We don't choose where we're going to do the sampling, we do it randomly. So to make sure our estimate is representative, so isn't just in the clumps or just in the place where there's no clumps, it's everywhere. And to remove bias, we use random sampling. So to do that, we use a wireframe called a quadrat. The one that you used in school was probably about 1/2 metre by 1/2 metre. It was probably a white wireframe and separated into 5 columns and 5 rows. So this is called a quadrat. Now we've said that we need to remove bias by putting it down randomly. So to choose where to put it down, we need to set out a grid on our sample area. And we do that by laying 2 tape measures. So if this is the 0 on the tape measure, that might be 1, 2, 3, 4, 5, 6, 7, all the way up to 20 metres, and the same on the y-axis. 1, 2, 3, 4, 5, 6, 7. So we lay out 2 tape measures. We then need to choose random coordinates. A coordinate, remember, are 2 numbers, the first one being how far we go along the bottom axis, the second one being how far we go up the y-axis, and that's where we put our quadrat down. For example, if our first coordinate was 0, 2, and we go along 0 and up 2, so the quadrat would go there. If our next quadrat was 3, 1, we'd go along to 3 and up to 1, and we'd put our quadrat there. Okay, so you need to choose points, sets of coordinates where your quadrat is going to go down. This is a common 6-mark exam question, writing a valid method. So the first thing we do is set out our grid. Set out a grid using 2 tape measures. The next thing we do is we generate 10 pairs of random coordinates. We could use a random number generator in our calculator or a website. We could roll a dice, anything that removes bias. So use a random number generator to produce 10 sets of coordinates. The key is random. And the reason we're doing that, remember, is to remove bias. We've said that we want to do 10 repeats. Okay. I'll write it again, do at least 10 quadrats. And you're going to count how many plants lie within the boundary of that square. Are in each quadrat. So each time you put it down, you count how many daisies are in it, or dandelions, whatever it is you're counting. We now need to calculate an average. All right. So we're going to calculate an average by adding up however many quadrats we've got and dividing by how many there are. So if we did 10, we'd add up all 10 and divide by 10. So when average, I mean a mean, really. So calculate a mean. And then here's the thing that they try to catch you out with. This quadrat is 1/2 metre by 1/2 metre, which means in total that quadrat isn't 1 metre squared, it's only 1/4 metre squared. So if using a 0.25 metre squared quadrat, then times your answer by 4 to make it as if it's 1 metre squared. So a whole metre along there and a whole metre up there. Then multiply by 4. Last thing you do then is times by the area of the field or the sample area. So you multiply your mean or your adjusted mean by the total area of the field. We don't always want to sample in a square. Sometimes we want to find out how things change along a line, and for that we use a transect. So this is when we sample along a line. Often it's to measure a change along that line, so up a hill or down a hill or past a tree, where there'll be a change in light intensity. Often to measure a change. Let's draw what that might look like then. So here we've got a tree. Here we've got the sun in the south. And for some of the day, that casts a shadow on this area of grass. So if we want to see the effect of light intensity then, and this is our grass along here, then we would lay our tape measure down in a straight line away from the tree. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So that's our tape measure. And this time, instead of doing one upwards as well, we're just going to put the quadrat down. We could either do it every metre, which would be called a continuous belt transect, or we could do it every other metre or every 5 metres, depending on the distance we've got to go. I'm going to draw it as if we're putting the quadrat down every other metre. So there's the first one, the second one, the third one, the fourth one, fifth one, the sixth one, etc. So this is the shadow, and these are the quadrats. As well as counting the number of plants, whichever species it is that I'm interested in, I'd also take a light meter reading. So I might want to use a light meter. It's often got a sort of light sensitive thing there, and then a dial or a number that tells you how strong the light is. So in this particular case, I'm looking at the effect of light intensity, so I'd also need to measure the light intensity at each quadrat too. Measure light intensity with a light meter. All right, so again, we could get asked a method on this. So number 1, you'd lay out your tape measure. Along whatever change it is you're doing. In this one, it was light intensity. It could be going up a hill. It could be going across a footpath, looking at the effect of trampling. So along the change. Number 2, you're going to put a quadrat down every 5 metres or every 2 metres. I've done it every 2 metres. Number 3, the one that everyone misses out, you need to say that you're going to count what's in the quadrat. That was the most common missed out answer a couple of years ago. Count the number of plants, or whatever it is you're measuring, in each quadrat. You also need to measure whatever it is you think is changing. So in this case, it could be light meters. If I was doing soil pH, I'd have to take a soil sample at each quadrat. And then lastly, to see if there is a relationship, I'd plot a scatter graph to see if there's a correlation between light intensity and number of daisies, for example. And that's it. Well done. You know what your job is to do now. To go away, learn this, and have a go at some exam questions. Well done.